Okay, good morning everyone. Happy Friday. So, welcome to part 2 of the nervous system, right? And in yesterday's lecture we talked about the structure of a neuron, and this was the generic structure of a neuron. There are subtypes which we won't get into. But today the main focus will be how does a neuron function, how does it get its input, how are the signals being transmitted. So that would be the focus of today's lecture. But before we get into that, I hope you remember that I ended class a minute or two early because I saw the facial expression was like, get us out of here. So I didn't finish everything I wanted to tell you about the axoplasmic transport. So what I was telling you is that when proteins are synthesized in the cell body, in the neuron, we have proteins being transported from the cell body all the way to the synapse, right? And the way that happens is through axoplasmic transport, right? And so axoplasm is the cytoplasm within the axon. And the way these materials will be transported from one end of the neuron to the other is through the macrotubules, and on the macrotubules you have the motor proteins which are going to take steps one at a time, and that uses ATP. And so I told you that we have kinesin which will move towards the plus end, which means moving away from the nucleus towards the cell membrane, and then you have dynein which is minus end directed, that helps to move vesicles filled with proteins in the opposite direction. And the movement towards the plus end is called the anterograde transport, and the movement towards the minus end is called retrograde transport. And I'll just mention something which is interesting. It's not on the slides, but it's interesting to know that this retrograde transport is what is used by the rabies virus to pass on, from a site of infection all the way to the central nervous system, where that's when retrograde transport is used. But in general, we'll talk about anterograde transport, and there seems to be two different speeds at which proteins are transported. So there's this fast transport which happens around 200 to 400 millimeters per day, and when they looked at the proteins which were transported at this fast rate, they were those which were produced in the rough endoplasmic reticulum. So these were proteins which were transmembrane proteins, or those which were secreted, lysosomal proteins. So whatever would be synthesized in the rough endoplasmic reticulum, they were transported in this fast mechanism. On the other hand, when we looked at the slow transport proteins, they were the cytoplasmic proteins which are produced on the free ribosomes. And I want to briefly tell you some of the experiments which were used to determine that the proteins were traveling at two different speeds along the axoplasm. And so they used rabbits, and they injected radioactive amino acids into the retina, and they would wait. So they had many rabbits which had these radioactive amino acids injected, and they would wait and they would sacrifice these rabbits at different time points. And what they were trying to do is they were trying to see how the amino acids, if they are injected in the retina, they are going to travel along the optic nerve. And so the cell body would be in the retina, and the axons would be passing through the optic nerve. And so by taking slices along the optic nerve and seeing where the proteins were moving, they were able to come up with this set of data. They saw that in this graph you have radioactivity shown on the y-axis, and the distance covered as time elapses shown on the x-axis. So they found there were two distinct peaks. One peak was moving slowly, around 0.5 to 2 millimeters per day, and the other peak was moving pretty fast, around 200 to 400 millimeters per day. So this seemed to indicate there may be two different mechanisms by which this transport was happening. And what they found out later was that it is not two different mechanisms, it is still the same mechanism where you have the vesicles carried by the motor proteins, and they're taken step by step. But it was found that sometimes these vesicles, instead of being moved continuously along, they would take a break in between, and then they would continue moving. So the analogy for this would be like, let's say you're driving on the highway, you stop at a rest station, you may take a half an hour break, and then you continue moving again. And so the question is, what was causing these proteins to take a break in between, and rather than just moving along the highway? And so they found that there were these proteins known as catechin proteins, and so it's named after katana, the samurai sword, and these were Japanese scientists who discovered these proteins. So they were capable of cutting out rafts of the cytoplasm and then causing it to be moved along the microtubules, and sometimes the proteins would get incorporated into the cytoplasm, and that's why it would be thought to be stopped in between. So is this clear why there was a continuous transport and the discontinuous transport of proteins along the cytoplasm? So in the continuous transport, it's just going along the microtubules carried by the motor proteins, and the discontinuity is caused by proteins being integrated into the cytoplasm, and then after some time they are cut by the catenin proteins and then transported again. So with that, we will begin with an overview of what I hope to talk about today. So we will be talking about the glial cells. So these are other cells in addition to the neurons, whose main goal is to support the neuron function. And we have around 4 types of these cells in the central nervous system and 2 types in the peripheral nervous system. So we'll talk about that, and then we'll move on to talk about the membrane potential. So I told you that the way the nerve cells can communicate with each other is by the movement of ions across the cell membrane, and that will lead to what's known as membrane potential. And so we'll talk about this concept, and then we find that action potentials are how the nerve impulses are sent from the cell body all the way to the synapse. So we'll see how action potentials are generated, and eventually we'll see how they can be propagated along the axon as well. And when the action potential reaches the synapse, it will lead to synaptic potentials, which will thereby cause the release of neurotransmitters. And these neurotransmitters being released will serve as the input for the next neuron. Okay, so I believe we'll get onto action potential generation in today's lecture, and then we'll pick up with the other topics on Monday next week. So let's start with the glial cells, which are found in the central and the peripheral nervous system. So notice here we have a cross section of the spinal cord, and the spinal cord is part of the blank nervous system, peripheral or central? Central nervous system, right? And so you notice that there is this gray matter in the middle, and then there's white matter around it. And we are going to look at a cellular level what is causing the structures that appear as gray matter or white matter. So let's look at something which we are already familiar with. So this blue part here indicates the cell body in the neuron, and that cell body is going to give rise to the axon shown here in blue. But you notice that these axons are surrounded by a myelin sheath. That's what looks in this light yellow color. This is the myelin sheath. And where that myelin sheath arises is from these cells known as oligodendrocytes. So here's an oligodendrocyte. And you notice a single oligodendrocyte can have these myelin sheaths forming around multiple axons. Okay. And we also find there are these star-shaped cells called astrocytes. They are interacting not only with the neuron cell body here, but also it is forming this sheath around a blood vessel. So here's a blood vessel, and it's forming a wrap around it, and that will serve as the blood-brain barrier. And there are also these smaller cells called microglia. These are the immune cells found in the central nervous system. So it will help to phagocytose the debris or small pathogens, and so they are the immune cells in the central nervous system. And finally, notice that shown here in pink, we have these ependymal cells. They are cells which produce the cerebrospinal fluid, which will pass through the central canal of the spinal cord. So this is just an overview of what cells you would see in the central nervous system. And notice that wherever you have the neuronal cell bodies, that's where you have the gray matter, whereas where you have the myelin sheath, that would be the white matter. So the gray matter is where you have the input coming in and the input is processed, whereas the signals will be sent to the white matter. So overall, we can say that the glial cells, they're going to support and protect the neurons, and they also will make up more than half of the volume of the nervous system. And there will be specific types of glial cells for the central nervous system and the peripheral nervous system. So what we just now did is have an overview of what are some of these cells, and now we'll get into the details of each of those. So this is an overview slide showing you that you have the satellite cells and Schwann cells in the peripheral nervous system. They are the glial cells of the peripheral nervous system. And in the central nervous system, we will have astrocytes, ependymal cells, oligodendrocytes, and microglia. I briefly told you what microglia is, and now we will talk about the remaining three in the upcoming slides. So let's first talk about astrocytes, and I've just pointed out two astrocytes here. And they have these large cell bodies with many dendritic projections, and that's what makes them look like a star, and so the name astrocytes. And the main function of these astrocytes is to form the blood-brain barrier. Keep in mind that the substances inside the blood, like white blood cells, cannot enter into the nervous system. If it does, it could cause an autoimmune response. And so there is this blood-brain barrier which will control what can move from the blood vessels into the nervous tissue. And what is causing the seal between these astrocytes are blank junctions. Whatever junctions do you think will form this barrier? Tight junctions. Thank you very much. It's very encouraging to hear a quick response. So we also find that it's going to cause a three-dimensional framework for the central nervous system. And these astrocytes will also repair any damaged nervous tissue, and they will even guide the development of neurons. We won't get into neuron development, but know that astrocytes play a role in that. And the interstitial environment. So the interstitial fluid is the fluid which surrounds the cells and also the blood vessels. That environment will be controlled by the astrocytes. And also these neurotransmitters, which will be released from in the synapse to communicate between the neurons, that can be recycled by these astrocytes, and we'll briefly talk about how astrocytes do that when we come back on Monday. So having mentioned that about the astrocytes, we can move on to the ependymal cells. So the ependymal cells, they will line the ventricles of the brain. So in the brain you have these large cavities which are called ventricles, and also along the central canal of the spinal cord, the ependymal cells will line that, and they will produce the cerebrospinal fluid. And this figure at the bottom is just showing you how the cerebrospinal fluid is flowing through the ventricles in the brain, and it's also going to cushion the brain so that it's not directly touching the skull. And so we see that this cerebrospinal fluid is going to cushion both the spinal cord and the brain. And because the brain is floating in the cerebrospinal fluid, its effective weight is a lot less. So it makes the brain buoyant. And you know that when you're in water, you're a lot lighter than when you're outside the water. So we find that the brain and spinal cord are lighter. And also there is nourishment and waste removal which can happen through the cerebrospinal fluid. And so it helps to maintain the homeostasis of the interstitial fluid in the brain, and also that which surrounds the spinal cord. So with that, we'll move on to the oligodendrocytes, which is the third type of glial cells. Notice that we find these oligodendrocytes, as I mentioned, they will form the myelin sheath around the axons in the central nervous system. So in the peripheral nervous system, you have other types of cells forming the myelin sheath, but in the central nervous system, it's the oligodendrocytes. And we find that this will provide a structural framework for the axons, but more importantly, it's going to cause the nerve impulses from the neuronal cell body to move at a much faster pace along the axon. So on this image to the animation to the left, notice that when it's unmyelinated, the signals are passed along very slowly, whereas when you have the myelin sheath, it will help it to move along a lot faster. The exact mechanism of how it carries this out, we will talk about when we come back next week. All right. So with that, we move on to the glial cells of the peripheral nervous system, and there are only two types of cells we consider here. The first one are satellite cells, and they will surround the ganglia. And what we mean by ganglia are these clusters of the neuronal cell bodies. It's not the axon, but just the cell body is going to be surrounded by these satellite cells. And these satellite cells have a similar function to astrocytes. They will regulate the interstitial fluid, and they also help to support the neurons in the peripheral nervous system. Moving on to the second type of cells in the peripheral nervous system, the glial cells are Schwann cells, and they're also known as neurolemocytes. So either name is okay. And these Schwann cells will form the myelin sheath around the axons. So we find that the satellite cells will surround the neuronal cell bodies, whereas the Schwann cells, they're going to form a sheath around the axons. And let's see how this sheath is formed. One more important thing. We find that only a single Schwann cell will myelinate a single axon, whereas one oligodendrocyte can myelinate multiple axons, whereas a single Schwann cell will myelinate a single axon. So the way this happens is, assume that this hand here is the axon and this other hand here is the Schwann cell. It's going to form a groove and wind around it, and then this cytoplasm is going to form multiple circles around the axon. And notice that as these the Schwann cells wind around the axon, the amount of cytoplasm which is being left within this Schwann cell is very limited. So as the Schwann cells wind tightly around it, it's going to basically have a bunch of lipid bilayers which are surrounding the axon. And these lipid bilayers will form an insulating material around the axon, and that's part of the reason why the nerve impulses can be moved pretty quickly when the axon is myelinated. All right, and we'll talk about this mechanism on Monday. So with that, we move on to the concept of membrane potential. So in this cartoon here, you'll notice that you have the plasma membrane shown here in blue. It's made up of this lipid bilayer which has phospholipids, and then the extracellular fluid is shown here in purple, right? And the cytosol is shown here in yellow color. And we find that you have a lot of charged substances both inside and outside the cell. So inside the cell we have these proteins which are shown in purple. They have a negative charge, and then you have more potassium ions indicated in orange are more plentiful in the cytosol, whereas in the extracellular fluid you have more sodium and chloride ions. So the key here is you have charged substances on both sides of the membrane. And the membrane potential is the measure in the difference of charge across the membrane. So I repeat, the membrane potential is the measure of difference in the charge across the membrane. And the way membrane potential is expressed is in terms of millivolts. Right? So it's going to be expressed in terms of what's the charge difference with respect to inside the cell versus the outside the cell. So if you notice this voltmeter here, it's indicating that the membrane potential is around negative 70 millivolts, and that means that the inside of the cell is more negative than the outside of the cell. Alright, so I'll be clear what is membrane potential and how it is expressed. Yes? Okay, now we can move on to what can change the membrane potential. Right? So whenever you have the movement of ions or these charged particles across the membrane, the ions move into the membrane or they move out of the membrane, it's going to change the membrane potential. Okay? So membrane potential can be changed by the movement of ions. So the next question is, what can cause these ions to move either into the membrane or into the cell or out of the cell? So we find that you have passive ion channels, and these are also called as leak channels. And leak channels are transmembrane proteins which will always be open, meaning that whatever happens, those ion channels will permit respective ions to move in and out. So here in this cartoon we have in orange depicting a leak channel for potassium, and here we have another leak channel for sodium in purple. So ions can move past these leak channels, and that can change the membrane potential. And secondly, one more thing is, notice that it says it's passive, meaning that there is no energy required to cause these ions to move across. It's mainly the concentration gradient which will push the ions across one side or the other side. Secondly, we have these active sodium-potassium pumps. There are other pumps too. There's an example of an active pump, and it's a sodium-potassium pump. And it's said to be active because it needs ATP in order to function. And why it needs ATP is it's going to move ions against the concentration gradient. It's doing the opposite of what the passive or the leak channels do. It's moving ions against the concentration gradient. So notice here that it's going to move sodium ions out of the cell, right? And we already have a high sodium concentration in the extracellular fluid. So it's moving sodium ions out of the cell against the concentration gradient. And simultaneously, it's also moving potassium ions into the cell against the concentration gradient because there's already a high potassium concentration in the cytosol. Now the third thing which can cause ions to move are active ion-gated channels. And these are channels which need to be opened by something. It's like you need a key to open this gate. And the key for opening this gate could be three types of keys. You could have a chemical key or a ligand. So I mentioned briefly in our discussion yesterday that you have many types of input. Right? You could have the input from your sense of sight, sounds, smells, taste, touch. Right? So among those inputs, you could have like a ligand like acetylcholine, right, binding to a receptor, and that acts like the key that causes this channel to open, and as a result, ions can flow in. When you take away the chemical or the ligand, the channel will close. Right? So we have these chemically gated ion channels. Secondly, we also have voltage gated ion channels. And what causes these channels to open is a change in the voltage, or in other words, as a change in the membrane potential. So notice here that in this very first panel, they're showing the voltage gated sodium channel, and at rest, we find that these channels are closed, and so no sodium can flow in. But notice that in the second panel here, the voltage changes from negative 70 to negative 60, which means that it's become less negative. In other words, it means it's become depolarized. And what causes this depolarization was the inflow of positive ions in the nearby vicinity. And what caused that inflow of positive ions was some stimulus, right? Could be any one of those 5 stimuli here: smell, touch, taste, whatever. And so as this membrane potential changes, the activation gate opens and sodium ions can flow in. But keep in mind that there's also an inactivation gate which is not dependent on the voltage. The inactivation gate will open and close stochastically or randomly, right? So this inactivation gate closes or opens randomly, and by the time the positive ions come in and it reaches the membrane potential of 30 millivolts, it can just randomly close, and it would take a while for that inactivation gate to open again. And this is a pretty important concept. That's why we're talking about it a second time today, right? We talked about it once yesterday, we talked about it a second time today, and we'll talk about it a third time later in the lecture when we discuss the action potential. So the third type of active ion channels are mechanically gated channels, and this concept is quite simple, right? You have the membrane which is not distorted and it's closed, but when there's touch receptor being activated because of mechanical distortion, we see these channels will open and ions can flow in. You take away the touch stimuli and the channels will close and positive ions can flow in. So take home message in this slide is what causes ions to move across the membrane. It could be a number of things. It could be passive channels, lead channels which are always open. It could be active pumps which use ATP and move things across the concentration gradient, or ions can move because a specific gate is opened by either a chemical or a change in voltage or by mechanical pressure. Yes, what's your question?
Is it important that we distinguish between a channel being closed and a channel being inactivated?
Absolutely. And I'll come back to that. The question was, is it important to know whether a channel is closed or inactivated? And I said yes. And because depending on whether it is, so the bottom line is, are ions flowing across it, right? Are the ions not flowing in because the activation gate is closed? Are the ions not flowing in because the inactivation gate is closed, right? So we have to differentiate that, and it will tie into a concept of refractory period. So yes, so if this is not clear, we will be revisiting it again. So are there any other questions? Yes?
If it's negative 70
millivolts,
that means it's more negative in the cell than outside.
And then when it gets to plus 30 millivolts, is it more positive or negative inside the cell? It's more positive, right? So, okay. Another question before we move ahead. What opens the activation gate in the voltage gated channel? What is it? A stimuli which does what? Which causes depolarization. Thank you very much. So the opening of the activation gate is due to depolarization in the close by vicinity. Second question. What causes the inactivation gate to open or close? Random, right? It's random. It's stochastic. It's going to randomly open or close. Okay, so now that we've talked about membrane potential, we will encounter three types of potential in a neuron. So the first one is called resting membrane potential. The second is a graded potential. And the third one is the action potential. So just a heads up, we'll revisit this again. The graded potential is what can eventually lead to a action potential. I repeat, a graded potential is what can eventually lead to an action potential. So let's go through each of these. So resting membrane potential is the membrane potential of an unstimulated resting cell, right? So there is no stimuli coming in, the cell is at rest, it's in at the resting membrane potential. And notice in this cartoon here, it shows that the membrane potential is around negative 70 millivolts. And the reason for this is that all of the gated channels, which we talked about previously, mechanically gated, chemically gated, voltage gated, all of them are closed at this point, right? And that is the reason why the cell is said to be having a resting membrane potential. And as this cartoon shows, notice that the sodium channels are closed, the potassium channels are also closed. Now we move on to the graded potential. So graded potential is a temporary localized change in the resting membrane potential. So let's work this starting with the words change in resting potential, right? So it's going to change from this negative 70 millivolts, the membrane potential, but that change will be localized to a very small area, and that change will be temporary, right? It's going to be just a passing change in the membrane potential. So that's what a graded potential. So what causes a graded potential? It's going to be caused by a stimulus, and the stimulus could be any of the 5 senses being activated: touch, vision, smells, and so on. And we find that the stimulus is going to arrive in the input region of the neuron. So it's going to arrive in the neuronal cell body through the dendrites, right? So the dendrites will receive a signal, and that's how the graded potential is going to be reaching, right, the neuron in the input region. So now we move on to the third one, and that's the action potential. And the action potential is an electrical impulse, and it's produced when a graded potential causes a threshold to be reached. I repeat that an action potential is produced when the graded potential is going to cause a threshold potential to be reached. So notice that as the stimuli is coming to the input region, when we reach a certain threshold, it will cause the action potential to be triggered. So let's look at what this means. And once the threshold is reached, the action potential will be passed along the axon. And I will explain what we mean by this threshold in a couple of minutes, but just bear with me, right? And once this action potential is triggered, it's going to move all along the axon in the conductile region. So the graded potential happens in the input region, whereas the action potential will pass all along the conduction region. So let me give you a real life analogy, right, of how I think about graded potential versus action potential. So you guys have heard about these competitions where people sing on TV. What is it called? American Idol or Britain's Got Talent, whatever, right? I don't watch those shows, but I know enough to realize that before you get to show up on TV, they have this audition, right, which happens, and there are some funny videos on YouTube where people have terrible voices. They show up for the audition and they're really upset that they don't get selected. Okay, so think about that scenario. And a graded potential is where you show up for the audition and you sing, and they're like, okay, well, we're giving you a grade, right, on your performance, but it's not good enough for you to pass on to the next level, right? So the outcome of you showing up was temporary, and it is very localized, right? It's just in the audition room, and then unless you do something stupid and it goes viral, right? So it's a very localized occurrence happening in the audition room. But let's say you do a very good job and your performance reaches a certain threshold, then they're going to send you all the way up to the next level, right? That's when the action potential is activated. Yes?
Could you also think of this as like a foreign body that gets to a valve, and then if there's enough of a situation where the valve gets turned, it just goes through there?
Absolutely. If that analogy works, if you're physics majors and or engineering majors, you can use that too. Right? So a threshold has to be reached, and I'll just give you a heads up. The threshold is referring to a certain membrane potential being attained, right? And that can open a voltage gated channel. So now I want to spend a few minutes talking about resting membrane potential, then we'll talk in more detail about the graded potential, and finally we'll get to the action potential, right? So we see that the resting membrane potential is going to exist because of 3 things. Okay, the resting membrane potential in the unstimulated cell at rest is existing because of 3 things. The first is that you have a difference in the chemical and the ionic composition between the extracellular fluid and the cytosol. Repeat, you have a difference in the chemical and the ionic composition of the extracellular fluid and the cytosol. So what are these differences? First and foremost, notice that I have put a red box around the sodium ions. So you will have more sodium ions in the extracellular fluid compared to the cytosol. That's the first thing. Secondly, we find that the potassium ions will be at a higher concentration in the cytosol versus the extracellular fluid. And thirdly, we also see that there are chloride ions which will be more abundant in the extracellular fluid. So in short, we can say that there is a difference in the composition of the cytosol and extracellular fluid, both in terms of ionic and chemical composition. Secondly, we find that the plasma membrane is selectively permeable. And I mentioned the term selective permeability, I believe, in lecture 2 when we talked about diffusion and properties of water. Here is the actual application in the cell of a selectively permeable membrane. And what we mean by selectively permeable membrane is that some substances can get across the membrane but others can't. And so what are these? So notice that potassium ions will have more leak channels than sodium ions. I repeat, potassium ions have more leak channels than sodium ions, and so potassium can easily leave the cell, or can even come into the cell if it wants to. But you have a higher concentration of potassium inside the cell, so it can easily leak out. And thirdly, we find that inside the cell you have a number of charged proteins, and these proteins are negatively charged, and they are too big to get past the membrane. And also because they're charged, they can get this hydrophobic lipid bilayer, so they're going to stay inside the cell. So it is these three things which will eventually establish the resting membrane potential. So I just want to briefly talk about how this resting membrane potential will relate to Ohm's law. Right? Ohm's law says that the electric current I is equal to V by R, voltage by resistance. And let me tell you how each of these parameters would relate to the neuron, which we talked about in the previous slide. So voltage is the force that moves the current, and this will be determined by what is the intracellular concentration versus the extracellular concentration of specific ions. So if you have a greater concentration of certain ions outside the cell, you want to come in, the force will try to move it inside. And current is the actual movement of the charged particles or the ions across the membrane, right? And what can cause the movement of these ions across the membrane is there have to be channels which permit these ions to get past the membrane. And resistance is if you have how much of the membrane is going to resist the flow of ions. So if you have more open channels, you will have low resistance. If you have fewer open channels, it will cause greater resistance to the movement of ions. So again, just notice your leaky membranes will have a low resistance and they allow more movement, whereas if there are fewer channels, you have high resistance. Keep in mind that this myelination, which I told you previously, caused by either the oligodendrocytes or the Schwann cells, they will lead to high resistance. And the reason for that is when the myelin sheath is wrapped around the axon, you don't have any of those voltage gated channels which can open and close, because it's like you have a tube with holes in it and you take a tape and you wrap it all around those holes. So those holes are non-existent. Okay, so now we'll move on to talking about an important concept called electrochemical gradient. Right? I'm going to tell you what is electrochemical gradient, how is it determined, but the take-home message in that is once you understand the electrochemical gradient, you will be able to say why potassium ions will move out of the cell, whereas sodium ions want to move into the cell. Okay, so the goal of talking about electrochemical gradient probably for the next 10 minutes is to tell you why sodium ions want to move out, but potassium ions want to move in. So the electrochemical gradient, it's got two parts of the word, electro and chemical. And so we will see that the electrochemical gradient is determined by two things. It's determined by the chemical gradient and the electrical gradient. So chemical gradient is saying that it's the passive movement of ions across the plasma membrane driven by the concentration gradient. Right? So the key here is the word passive movement, meaning that it's moving across a leak channel. Okay? Passive movement means it's moving across a leak channel, and what is driving the movement of the ions is the concentration gradient. Whereas the electrical gradient is the movement of ions across a leak channel, and it's going to be mediated by the difference in charges. So if you have a higher concentration on one side of the membrane, it will move from high to low concentration, whereas if you have a lot of positive charges on one side of the membrane and the inside is negative, those positive charges will be attracted to the negative charges. So the electrochemical gradient is going to be a sum of the chemical and the electrical forces acting on the membrane. Okay, so let's take a look at the electrochemical gradient for the potassium ions. So I have 3 points here: chemical gradient, electrical gradient, electrochemical gradient, and in this picture here I have 3 arrows. All right, and each of these arrows is going to depict one of these gradients. So the orange arrow here, it says it's the potassium chemical gradient. I notice that it is actually pointing out of the cell. It's saying that ions should move from inside the cell to outside the cell. Okay, whereas this white arrow, it's denoting the electrical gradient, and it's saying that ions should move from outside the cell to inside the cell. Right? And you'll notice that this orange arrow is slightly thicker than the white arrow, which means it's slightly stronger, and so they will sort of almost cancel each other out. But notice that there is a slight force which will push the potassium ions out of the cell because this chemical gradient is apparently stronger. So just by looking at this picture, you can tell me that electrical gradient will force it out of the cell. Chemical gradient says go out of the cell. Electrical gradient says come into the cell, and you see the combined effect. But what you can't explain is the reason why, right? And the rationale is what I want to explain now. So if you look at the potassium ions, right, they want to go out of the cell because there's a higher cytosolic concentration of potassium ions. And so that is why the chemical gradient for potassium says, please go out of the cell. It's going to move down the concentration gradient. Are we clear on that? Yes? Okay. So let's move to the electrical gradient. Why do the potassium ions want to come into the cell? This is caused by 2 reasons. Reason number 1, we find that the positive charges outside the cell will be attracted to the negative charges inside. Right? We're straightforward on that point. Second point is that these positive ions which are outside the cell, they are going to repel each other. Right? So they rather not be close to each other, and they will be forced into the cell. Is that clear? Why the potassium electrical gradient is forcing it inside the cell? Because it's attracted to the negative charges, and the positive charges will repel each other. And so we find that when you take the sum of these 2 forces which are acting in opposite direction, we can say that the potassium chemical gradient is weakened by the electrical gradient. So I hope you can understand why this orange arrow becomes very skinny, right? Because it's being weakened by the electrical gradient. Now let's move on to the other example of sodium ion gradients, right? So in the sodium ion gradient, we notice that the chemical gradient is pushing it inside the cell, and the electrical gradient is pushing it inside the cell. And so both of these gradients will combine together, and you have a great push to move sodium ions into the cell. Right? So what is the rationale for all of these 3 arrows? So we find that the sodium ions are moved into the cell due to the higher concentration of sodium in the extracellular fluid. Okay, that's pretty straightforward. And then moving down to the electrical gradient, we find that sodium ions are driven into the cell due to the same 2 reasons mentioned for potassium as well. The positive ions are attracted to the negative charge inside the cell, and the positively charged sodium ions will repel each other, and they'll be happy to cross the membrane. And so we find that the sodium and the sodium chemical and electrical gradient will drive it into the cell. So if I went back to the previous slide and asked you, why does sodium want to move out of the cell, and why does sodium want to move into the cell, and why does potassium want to move out of the cell? Can you explain it using this thing here? Great. So we find that this is a form of potential energy, right? Where you have like a dam with water behind it, and even a small opening will cause the water to be forced out. And so even a few sodium channels opening will cause sodium to be forced into the cell. Even a few potassium channels opening will cause potassium to be forced out of the cell. Okay? So we have 3 minutes, and I want to talk to you about a concept known as the equilibrium potential. So the equilibrium potential is the membrane potential at which there is no net movement of particular ion across the cell membrane. Right? And so this figure on top is showing you that the first panel we've already talked about in detail. But if you look at the second panel, we see that the potassium chemical and the electrical gradient are balanced. And so there's going to be no net movement of ions across the membrane. And if you look down here at the sodium ion gradients in the second panel, you notice that again the chemical and the electrical gradient are balanced, and so there's no net movement across the membrane. Now what caused these two forces to be balanced? It was the opening of either potassium or sodium channels, and ions were allowed to move until both of those gradients could be balanced. And you'll see that this equilibrium potential for sodium ions is around negative 90 millivolts, whereas for the equilibrium potential for the potassium ions is close to negative 90, whereas for sodium it's plus 60. And so we see they're pretty far apart. And the reason for that is there are more leak channels for potassium, or in other words, there's a higher permeability to potassium, and that's why the equilibrium potential for potassium is closer to the resting membrane potential. Whereas you have very little permeability to sodium, and that's why it is so far away from the resting membrane potential. Let me just say one thing in passing, that the number of leak channels for potassium would be 95%, whereas only 5% of those leak channels would be for sodium. Yes, what's your question?
I was just wondering if this is part of the reason why too much sodium in your system is such kind of a pain in the butt compared to other things that you can have too much of.
Right, so the question is, having too much of the sodium, why is it pathologically related? Anything which changes the ion concentration, right, even which is sodium or potassium, concentration will change. It can affect muscle and nerve function, but we'll get into that. So we'll stop at this point, and I'll tell you that we can calculate equilibrium potential also using this fun equation here, which I will talk about when we come back on Monday. All right. But let me finish with one good news before you all leave. We will only look at this equation very superficially. If you want to get into the depths of the richness of this equation, we can do it in a more advanced course, right? But I will touch upon it briefly. Everyone